Hello, my name is Ferda Eser. Today I will talk about you the laboratory evaluation of insecticidal activities and phytochemical analysis of Marrubium astracanicum subspecies astracanicum against Callosobruchus chinensis and Callosobruchus maculatus. There are many pests that harm human food in general and have the potential to cause several diseases in human body directly or indirectly. Depending on the dimensions of the damage between pests and humans, pests can lead to serious problems, especially in food. In legumes, which are an important food source for humans, especially beans and chickpeas, there are many important harmful species that must be combated from the process of harvesting down to the table. Callosobruchus sinensis and Callosobruchus maculatus are two important pests of legumes that are stored in warehouses. The larvae of these two pests feed inside the seed, complete their life cycle, and emerge from the seed as an adult insect. Studies have focused on the synthetic chemical methods of controlling the two pests that can cause serious damage to legumes. In recent years, the importance and use of environmentally friendly methods has been increasing. The usage of plant extracts is one of the new methods in biopesticide applications. Numerous studies have revealed that plants can be used for pest management. In this concept, Amasya is one of the important families and species of this family that have been used successfully in the control of very stored grain pests. Why we choose this plant? Let's talk about Marrubium astracanicum. The genus Marrubium that belongs to the Amasya family is an annual or perennial herbaceous species that are represented by nearly 50 species in the world and 19 species in Turkey. It is reported that some species of the genus Marrubium exhibit analgesic, antiprotozoal, and antimicrobial activities. The aerial parts of astracanicum have been used in Iranian folk medicine for stomachache treatment. The methanol extract of Marrubium astracanicum exhibits antibacterial activity against Staphylococcus aureus, Escherichia coli, and Enterobacter aerogenes. In the experimental section, first the collection of plant material. The aerial parts of the plant were collected from the Akta area in the province of Amasya in June 2018. The identification of the plant was performed by Professor Dr. Cengiz Yıldırım, and a voice specimen was deposited at the herbarium of 19 Mayıs University. For the extraction process, Marrubium astracanicum subspecies astracanicum, shortly MAA, was dried in the shade and crushed into small pieces with a grinder. The plant was extracted with methanol for 24 hours using the maceration method at room temperature. After filtration, the solvent was evaporated under reduced pressure to obtain the crude extract, which was suspended in distilled water and extracted with hexane using a funnel separator. The combined hexane layer was dried with anhydrous calcium chloride and then filtered. The hexane layer was concentrated by a rotary evaporator to produce hexane extract. The remaining layer of water was subjected to the same extraction steps using N-butanol to afford N-butanol extract, and the water extract was obtained after the lyophilization of the water layer at minus 80 degrees Celsius. How we made the rearing of seed beetle. All the undamaged chickpea seeds used in experiments were sterilized in an oven at 50 degrees Celsius for 24 hours and then cooled at room temperature. The adults of Callosobruchus chinensis and Callosobruchus maculatus were obtained from stock cultures grown on sterilized chickpea seeds. The adults of Callosobruchus chinensis and Callosobruchus maculatus were obtained from stock cultures grown on sterilized chickpea seeds. We use two different assay application. First is topical assay and the second is contact assay. For topical assay, a 2 microliter of liquid extract from each concentration of different plant extracts, hexane and butanol and water, was applied to the surface of the protonium of each Callosobruchus chinensis and Callosobruchus maculatus adult using a micropipette. For this purpose, 5 adults were placed in petri dishes containing 5 sterilized chickpea seeds, and the petri dishes were tightly covered with some tulle for beetles to be able to breathe. The controls consisted of paths treated with hexane and butanol and water solvents respectively. Adult mortality was observed at 24, 48, and 72 hours after treatment. The seed beetles that didn't respond after light poking with a fine brush were considered dead. For contact assay, the extracts of different concentrations including 62.5, 125, 250 and 500 ppm were sprayed onto filler papers using a nanospray device. For the control groups, hexane and butanol and water solvents were applied by spraying on the filter papers. The filter papers were dried for 10 minutes and then placed at the bottom of plastic petri dishes. After 5 adults of both species were placed in petri dishes, prepared for each concentration of the extracts, 5 sterilized chickpea seeds were placed in each petri dish, and the petri dishes were tightly closed with some tulle. The adult beetles in petri dishes were touched with a fine-tipped brush at 24, 48, and 72 hours after application, and unresponsive beetles were considered as dead. For determination of the main compounds of the active extract, we use GCMS analysis. The gas chromatography analysis of the hexane extracts was carried out on an age-linked HP model GCMS apparatus. MAA extract was dissolved in ethyl acetate and then filtered through a polytetrafluoroethylene membrane filter. The identification of the compounds was performed based on their peak area, and GC retention indices, NEST, and Woolley libraries were used for the identification of the components. Table 1 shows the LC50 and LC95 values after topical applications of Marrubium astracanicum, subspecies astracanicum extracts on Callosobruchus chinensis and Callosobruchus maculatus. When the LC50 values of hexane extracts were compared after 72 hours of topical and contact applications, it is seen that the dose required for the death of Callosobruchus chinensis adults in topical applications is lower than that in contact applications. The same situation is similar in the adults of Callosobruchus maculatus. A possible explanation for these differences in toxicity seen in these two pests may be due to the ability of MAA extracts to penetrate the bodies of these pests and the ability of the pests to metabolize these compounds. In both water and butanol extracts, close results were obtained with either contact or topical application methods for both pests. Table 2 shows the LT50 and LT95 values after topical application of Marrubium astracanicum subspecies astracanicum extracts on Callosobruchus chinensis and Callosobruchus maculatus. It was observed that the time required to kill the adults was shortened as a result of the increased dose of hexane extracts which were applied to Callosobruchus chinensis and Callosobruchus maculatus adults in topical applications. Similar results were observed for both pests in water and N-butanol extracts in topical applications. It is understood that as the dose required to kill the pests increases, the time required to kill the pests decreases. As a result, it is important to use the most accurate method and the most effective dose in the insecticidal studies of plant extracts. Table 3 represents the LC50 and LC95 values of Marrubium astracanicum subspecies astracanicum extracts on Callosobruchus chinensis and Callosobruchus maculatus after 72 hours of contact application. The contact effects of plant extracts on Callosobruchus chinensis and Callosobruchus maculatus adults showed that the dose required to kill Callosobruchus chinensis adults was slightly higher than the dose used for Callosobruchus maculatus adults. And the evaluation of contact efficiency for both insect species, the species responses to different extracts were determined by comparing the highest values for lethal concentration and the highest values for lethal time. Table 4 gives the LT50 and LT95 values of Marrubium astracanicum subspecies astracanicum extracts on Callosobruchus chinensis and Callosobruchus maculatus after 500 ppm of contact application. Considering the LC50 values of Callosobruchus chinensis and Callosobruchus maculatus adults at end of 72 hours, the most effective extracts on adults were found as hexane, followed by water and N-butanol sequentially. When the LT50 values are obtained after the treatment of 500 ppm plant extract with Callosobruchus chinensis and Callosobruchus maculatus adults were compared, it was determined that hexane extract had the lowest time required for lethality, followed by water and N-butanol extracts respectively. Figure 1 shows the chemical structures of the main compounds of Marrubium astracanicum subspecies of astracanicum hexane extracts. Phytochemical analysis of the active extracts yielded 32 components. It is reported in GC diagram. And the main components, longipinan, palmitic acid, hexahydrofarnesyl acetone, and neophytadiene were determined as the major components of the hexane extract. The most abundant compound is longipinan. It is 29.61%. Conclusions. In this study, the various extracts of MAA were screened in terms of their insecticidal activities, against Callosobruchus chinensis and Callosobruchus maculatus pests, and the phytochemical content of the active extract was determined. The topical applications using the MAA extract were found more effective than the contact applications against Callosobruchus adults, and the effectiveness of the hexane extracts was higher than other extracts in both assays. In addition, the importance of the application dose and application time were revealed, as well as the methods applied and the use of MAA extracts against Callosobruchus adults. It was concluded that the adult mortality rates of the adults increased depending on the increase in the dose of the applied extracts and or the increase in the exposure time. As a botanical insecticide, MAA extracts have proven to be very effective against Callosobruchus chinensis and Callosobruchus maculatus pests, and it's thought that it will shed light on both laboratory and field experiments in pest control. The phytochemical analysis of the active extract we said afforded four main constituents, including longipinan, palmitic acid, hexahydrofarnesyl acetone, and neophytadiene. The antifungal and cytotoxic activities of longipinan have been reported, while the insecticidal properties of the compound have not been studied yet. And neophytadiene is an important terpene and possesses larvicidal activities. And another literature review indicated that hexahydrofarnesyl acetone had allopathic and pest control potential. Neophytadiene is an important terpene and possesses larvicidal activities, antipyretic, analgesic, anti-inflammatory, antimicrobial, antioxidant, and repellent activities against insect pests. Synthetic chemicals undoubtedly protect the stored products from Callosobruchus species, but their uncontrolled overuse can cause health problems in humans, particularly insect resistance and degradation of the environment. Therefore, there is a need for environmentally friendly and reliable products such as plant-derived products which have the potential use instead of synthetic pesticides. So many plant-derived products such as plant extracts are actively used in pest management thanks to their insecticidal activities. In Turkey, the Marrubium species is broadly scattered in South Marmara, Black Sea, and Mediterranean regions at an altitude of 1,600 meters. Marrubium astracanicum subspecies astracanicum can be grown in small areas by applying tissue culture, cell culture, and vegetative reproduction methods for sustainable harvesting. It was reported that Marrubium vulgare, which was grown wild in a wetland and dry sandy soils, was cultivated in Lithuania and harvested for medicinal uses. So the use of MAA extracts can be effective for the pest control as pest management.